Phosphate fertilizer is a $78 billion a year business. And applying arbuscular mycorrhizal fungi once can replace phosphate fertilization forever because it's regenerative. And it's not all soils, it's not all plants, but it's a massive displacement. So in studies where there is phosphorus in the soil that's not bioavailable, this is how you make it available to plants, and this is how you replace phosphate. And so this is an incredible breakthrough, but it's what nature was already doing before fertilization, before fungicides, pesticides, herbicides, because all those kill fungi, and then tillage and the break it and then leaving it bare so that it bakes and UV and oxidizes, all of that. So when we're talking about arbuscular mycorrhizal fungi becoming regenerative, if you inoculate your plants, it's successful, and you only till 2 to 6 inches deep and then immediately plant seeds in that soil, then it will have those broken up roots around it. If you grow mushrooms and you're in the mycology world, it doesn't matter what kind of mushroom with the substrate. You feed one substrate and then you break it up and mix it with a new substrate, and then it eats that. And then, right? Yeah, that's how they're doing it. So if that's how mycologists are doing it. That's how mushroom growers are doing it. But we're doing the same thing if we are using tillage in between crops as a disturbance mechanisms to start our seeds. Now, planting immediately in it, the rhizospheric microbes, the chunks of root that have the mycorrhizal fungi in them, they will all transfer to the new roots, to the new seeds. It's so easy, it's so exciting, but it changes how people farm fundamentally. It gets away from the chemicals, gets away from just spraying everything with glyphosate to kill it for the season and dry it down. It's a different way of doing things. You're going to do cover crops. You're going to crimp it. You're going to be letting things mature, letting the soil recover. It's a path of fertility. And arbuscular mycorrhizal fungi, depending on the soil and other conditions, it can be up to 30 to 40% yield in addition to your normal yield. So it's a 30 to 40% boost in your overall biomass and yield every year. So for farmers, same amount of land, they're getting 30 to 40% more profit. Yeah. So it's a massive breakthrough for farmers right now that we've been able to have this technology with this Epifluorescence microscope that's LED. Now it's portable. Now it's safe. It's not a mercury-based lamp, you know what I mean? And we're not having risk for hazmat suit, issues everywhere. This is the power of this microscope, but it's also, I'm talking about the power of arbuscular mycorrhizal fungi. And so this is something that partners with over 90% of plants. There are non-mycorrhizals, like beets and Quinopodia family, like quinoa, and then amaranth, those kinds of things that are usually pioneer species, usually weeds, and highly oxidized and alkaline soils. So everywhere else where we normally evolved alongside these plants was more humid and more favorable soils and highly oxidized and alkaline, because that's where there was more water. And so naturally, where there's more water, it's more acidic. So most of our plants that we grow with are naturally partnered with that, and because it's naturally more acidic, it's more fungal. And so all those partnerships and those relationships in the soil that are displaced by like streptomyces and actinobacteria in alkaline oxidized, like that's the kingdom of fungi. As soon as you get to like pH 7 or more acidic, it's like, so arbuscular mycorrhizal fungi, over 90% of plants. It's almost all of our food. I mean, it's like, well, not blueberries, that's ericoid mycorrhizae, right? Not cranberry. I get it. There's a few, but everything else largely is partnering with the muscular mycorrhizal fungi, and has so many benefits. It increases nitrogen, it increases phosphorus, makes things that are not bioavailable plant available, and it's something we can see with this light, this epifluorescence lighting, this lumen unit. And so this is why there's a lot of confusion, I think, online, because there's a lot of interests and involved in this space, because farmers could be empowered, and then that means they're literally taking that profit from someone else, because they're able to just now not need those phosphate fertilizers, right? So it's really, and then it's also incredibly important to, like, when we're looking at some of the ways these things have been tested by universities, they'll spray glyphosate all over everything and then try AMF, but glyphosate prevents our muscular mycorrhizal fungi from growing. It's a fungicide. And so they're like, oh, it has no effect. And it's like, you just did that. And then there are some arguments that, like, phosphate doesn't autofluoresce. If you take a substance and dilute it and dilute it and dilute it and dilute it until its normal effects are no longer like apparent, or they don't happen, you can fool the science, you can mess around, but that's the thing, is it's like the way we're doing things and the way that people who are practicing this are doing it, you can just verify it. And everyone else can verify it. And everyone who's familiar with the space can immediately tell if you're trying to, like, pull a fast one. And that's why it's so powerful. And why so many people are getting involved. And there's a lot of excitement in this space, because people know, like, oh, this is the way out of this problem, no matter what, because we can see it. So phosphate, I'm going to show a picture here, show some video here. Phosphates, like triple superphosphate, this is it's this bag, and this solubilized changes the liquid to a green color, and then itself, when it's in crystals, it also has that green, that stereotypical yellowish green color. And so it is autofluorescing. It's undeniable. You can see it here. And you can copy this. You can do it yourself. And the thing that really shows you it is that, like, okay, here we can see the green background, right? And then here, where it's all dried up, it's black background because there's nothing there. It's all dried up. And it's all focused, the glow is focused where the crystals are. And we can see that these crystals are also on fungal hyphae. I've seen it in my work. And these crystals, fungi forms specific crystals, and they have specific minerals in them only. And so for them to all be consistently glowing, that means there's a common thread, and it's not calcium, it's not carbon, it's not hydrogen, it's not oxygen, it's phosphorus. And so when phosphorus becomes a phosphate or combines with other things, it seems to inevitably glow. And yes, you can dilute it down. And when we talk about things, when I'm saying phosphorus and a phosphate, phosphorus is the dominant element in a phosphate. That's why it has that word in it, and it sounds a little different. But same thing with, like, nitrogen and nitrate or nitrite. They're different forms of that element. And I think that there's some confusion online about when people say, like, the phosphorus cycle, and then they're like, oh no, there's a phosphate. It's not phosphorus. And so there's like people who are really confused about where these things come from. In regenerative soil, I go through all the elements that are plant essential, and we go over all of them. And that's why this stuff takes 20 weeks to teach. We got to cover it all and put it all in order. But I also wanted to make these videos because there's a lot of confusion online and a lot of misinformation on YouTube and Wikipedia and ChatGPT and some of the studies. You just got to see how they did it. You can't go off the abstract. You got to read their methods, and that's usually a section in the paper. And I like to see their pictures or their video. And the reality is a lot of their pictures are so terrible. It's like, so you can't ever actually tell if they were accurate. And that's on purpose, right? It's wild. But that's also why I have this imperative to test things. We also deduce. When I first started doing this work, I've looked it up and I found, like examples where phosphate does autofluoresce. You got to find the exact wording to get it to come out of Google Scholar. And then I also had peer reviewers, and I brought it all to them, talked with them about it. And so, like, that's the thing. It's like, you got to do all those steps. I've been doing all those steps because then it's replicable and people can do it. And because we have both the literature, and like I said, there's weird stuff out there, and misinformation out there, but you can so easily prove it to yourself just by doing it. So you can do this. And not only that, when we're looking at roots, you can see the pathway of things. So when it forms NADPH, which is a form of phosphate, that glows. But all phosphates are drawn into like the orthophosphates is the term we use for all. And so all the ones that are being bottled in, they draw it in like a straw, and we can see it here that it's concentrated in the middle like a filament. And so that's drawn like a straw through the hyphae and then delivered right to the membrane of the cell. And look, we can see here at the membrane of the cell, that's where that initial light happens, where that exchange is happening. And that's the beginning, and then it turns into a full-blown expression, the whole root, and that whole root glow is how you know that you've had a successful inoculation, a full inoculation. So it's rather simple. And then to know that there's been several different, and there should be several different successions. That's normal. Look at this. It's reddish. See how it's reddish? That's the hyphae that's senesced. That can reanimate next year when it touches new roots. But that, and it also can be absorbed by the plant, hypholysis. It absorbs that as stored energy in case of emergency. But that hyphae is there to reanimate or to be absorbed, but it's essentially dead or senesced or dormant. It only will reanimate if it's touching a new root. So this is their life cycle. You've just seen it. You've seen them initial where they start with, and the number one thing brassica mycorrhizal fungi accumulates is phosphorus, the number one element. And beyond the phosphorus debate, because it's obvious why they want it to be obscured and all that stuff, because it's a $78 billion industry. But we can just test it and see. And we can test our roots and see. With this technology, this lumen unit, I don't use dyes. I don't macerate it. I just take the whole root, and we don't crush it. We don't add water. We get to see things as they are. It's a fundamental leap forward, and it's so gentle, it's so powerful, and it's so fast. So everyone can do this. The glow is not entirely phosphorus. Some of it is a precursor to the phosphate in there, which is NAD and NADPH, which are the primary ones that are glowing. But they all are all indicative of phosphorus, because whenever there's that glow, even the proportion, the ratio of the precursor to the phosphate is always smaller, and it's always greater. And the final product is always the phosphate, which is the thing that fills it up. And so we have these incredible glowing roots that we can just go out and see, that we can verify. And the first part of it, the first part of that reaction where it's forming these phosphates is actually reduction of nitrogen. It's at the same time. It's double duty. So we know that you're gaining in nitrogen when you see that glow. We know that you're taking in tons more phosphorus, and you don't need phosphate fertilizer now. And so this is an incredible technology. And with mycorrhizal fungi being returned to the symbiotic state with plants that it originally had, that it originally evolved with, we simply are bringing our plants back online. And because of that, we no longer need that whole fertilizing, for the most part, that whole fertilizing industry. And we can have much more resilient plants. We can have that gain, that 30 to 40% gain every year thereafter. Not exponentially, just that gain stays because you kept the inoculation. So you can look at the inoculant before it goes in, make sure it glows. It's been proven over and over and over again, and it's established now that all parts of arbuscular mycorrhizal fungi autofluoresce. They've been arguing about what the glow is, because if you put 2 and 2 together 20 years ago, you probably could have displaced the whole industry by now, and peak phosphorus would have never been a discussion. But here we are, and we got to do it now, especially because they're talking about how it's going to go up in price. There's peak phosphorus, all this. No, there isn't. There's all this in a non-soluble phosphorus everywhere, and we just got to unlock it. And we can see it. We can prove it. We can prove these are the things that glow. We can do experiments. We can use the technology, and we can compare it with others and help farmers transition for the first time. Because when farmers can test the mycorrhizal inoculant and make sure that it's good, and then test that the inoculant took, so you have 80 to 100% inoculation rate every 1,000, every 10,000, maybe every 100,000 plants, depending on what you are. Maybe a ranch, right? That'll be every millionth plant, maybe. So we're going to be looking at roots. We're going to be making sure there's full inoculation, and then you come back the next year, and you make sure that it came back, and you get to say to the farmer, "You're regenerative. Your arbuscular mycorrhizal fungi is regenerative. All this is inoculated. You didn't spend any money, and you get all the benefit now. Keep it up, and it never will go away, because that's why it's here in nature. It's meant to stay." So I wanted to show you guys the value in this. I wanted to show you guys phosphate. I wanted to show you guys the crystals on the hyphae. I wanted to really reveal what I went through to figure this out. I wanted to know the colors. I wanted to understand it, and I went through the research. I went through the deduction, because you got to go through those elements and test them all out each. And then I went through a peer reviewing process, and now we can just share. We can just do it. And so this is a game changer. This technology changes everything, and you can join my course and learn about it. Or you can check out my book, Regenerative Soil Microscopy. The link is down below, and you can learn about it. I have it all in there so that you can start with just the book. But if you want to go deeper, you want to learn side-by-sides, all those kind of things, and also work with other people in a group setting. Work with people so that you see their slides, they see your slides. You end up seeing a dozen examples every week that we do the group sessions. And so we first do morphological and train your eye, and then we do group work for the second half. And so it's amazing. People learn so fast. And also, you figure things out, you get questions answered, you see what other people's setups are like, their cameras are like. Very quickly, you get to know everyone as well. It's an incredible community. I hope that you join us. Click the link below to learn more. I'm Matt Powers. Grow abundantly, learn daily, and live regeneratively. I'll see you guys soon. What if you could verify if your compost was actually doing its job? What if you could verify if the inoculants, the mycorrhizal inoculants, biofertilizers, are actually worth the money spending on them? What if you could diagnose what was going wrong with the soil or wrong with the plants and fix it? And exactly know what to address. It is all possible. And it's all things you can learn in the Regenerative Soil Microscopy 20-week online course that is starting September 25th this fall. Join us for the full in-depth course that pairs with the new book, Regenerative Soil Microscopy. If you want to know how to use a microscope to diagnose and evaluate your compost teas and extracts, your roots, mycorrhizal fungi, your soil, and all your amendments and biofertilizers, then this is the course for you. It's new videos at the start of every week for 20 weeks straight, and we have live Q&A at the end of every week. I'll send a survey out so everyone's questions get answered regardless of whether they're with us live, because I'll move the live around so that we'll reach people in different time zones because we're a global community. It's going to be incredible. There's live sessions that we're going to have with other people in the group with our microscopes. You're going to be able to see the work of other people on their microscope in real time and develop your eye. I'm going to be able to jump into your group, and we're going to be able to examine and troubleshoot and help everyone learn how to use microscopy to better understand their soil, their compost, their actual samples. We're going to do a lot of work together on interpretation and diagnosis and understanding and extrapolation from these tests. We're going to walk through it step by step together with me, the author and creator of the Regenerative Soil Microscopy book, course, and methods. So you're going to see going down to the original source material, why I chose what I did, the test results that I got that led me to the choices that I made to create the methodologies we have today. It will surprise you and inspire you because we are at the cusp of a revolution in soil science. So dig deep with us this fall in regenerative soil microscopy. Explore and understand the world at a completely new level and transform your soil, transform your plants, your food, your animals, your forage, your landscape, the environment from the soil up. And we'll cover everything. Not just bright field, we'll go beyond bright field. You've waited long enough. There's dark field, there's epifluorescence so that you can see the invisible fungi, so that you can see where phosphorus goes and glows. And also biological stains, so you can verify that things are actually active, and without the freezing, without the liquid molten agar. These are living samples that are just diluted 1 to 100 and then stained. It's incredibly easy. It's so fast, and it's an all new method. So I'm so excited about this. I've been able to verify things with world-renowned scientists, highly published scientists, people who have been teaching and focusing on this area of study of microscopy for 40 and 50 years. I am so excited to share this with you because we've all waited long enough to know how soil actually works, to not be fooled by the numbers and the math that has been legacied in. There is a path forward that disambiguates, that opens new doors, that pairs with DNA sequencing, that pairs with the new understanding of the way genetic transfer occurs, especially in the soil environment, the composting environment, and the rhizosphere environments. If you want to learn how to not just understand your soil, but to see that the things you're doing are actually working, that the money that you're going to spend or have spent was worth it, so you don't get fooled again. This is the pathway. We need holistic testing, we need holistic microscopy, and we need to combine them in a new methodology, regenerative soil microscopy. I hope you join us. I'm Matt Powers, author, educator, seed saver, citizen scientist, and family guy. Grow abundantly, learn daily, and live regeneratively. I'll see you soon.